Hi everybody, it's Dr. Hall, and we're going to get started with the first part of Lecture 2. So Lecture 2 is all about cells of the nervous system. On the first part, we're going to focus on neurons. So here's the outline for the entire lecture. So again, 2A, this first part is all about neurons. They're basic organelles that we're going to find, some unique properties, and some special features that we're only going to find in neurons, as well as a brief mention of the different types of neurons. And then 2B, the second part of lecture 2, is going to be all about neuroglia, which are the other types of cells that we find in the nervous system. So we have these two cell types, neurons and neuroglia. The neurons are kind of the superstars of the show. They are the cells that are sending and receiving information, which is the whole job of the nervous system, is to send, receive, and process information. So the neurons are really where it's at, and then the neuroglia are all the other cells. So if we're looking at this micrograph here, we can see in the center this large cell that has some projections going off of it. That is the neuron. And then all of these little, here's one, these little purple dots, those are the nuclei of these other cells, which are called neuroglia. Now, there are different types of neuroglia, as we're going to learn about. And as you can see, they outnumber the neurons by about 10 to 1. When these were first discovered, they didn't really know what they did. They thought, well, maybe they're just holding the neurons together, so they got named neuroglia. It comes from the Latin word for glue-glia. So they thought they're just holding everything together. We now know that there's different types of neuroglia. They do lots of different things, but that's where that kind of general term came from. So I like to think of it like the neurons are the celebrities, right? So they're the Taylor Swift, they're the Beyonce, and then neuroglia are the support cells that help them do what they're going to do. So it's their personal chef and their chauffeur and their agent and their housekeeper and whatever all else, right? So all the people that help the neurons to do their job. Okay, so now we're going to focus specifically in on neurons. Neurons are cells. So like any other cells, they are going to have all of the organelles that we're usually going to find. So quick review here. So the nucleus, which is the site of our DNA, which is what makes up our genes, that has the instructions for all of our cells for how to be parts of us. And then inside the nucleus is the nucleolus, which is that denser area. That's the part of the nucleus that produces RNA. RNA serves lots of different functions inside of the cell. Then there is the typo, rough and smooth endoplasmic reticulum. So rough endoplasmic reticulum produces and modifies proteins. Smooth endoplasmic reticulum makes lots of other types of molecules for the cell. Mitochondria, which are of course the powerhouse of the cell, they produce ATP, which is the energy currency for our cells. Golgi apparatus, which is really important in terms of, I think of it like shipping and receiving. It's really important in terms of taking things that have been produced by the endoplasmic reticulum and sending them off to where they need to go, packaging them up, all that type of stuff. And then the plasma membrane. So the plasma membrane is kind of the cell membrane that separates the cell inside from outside, right? Gives it its actual entity. And that's going to be important. So it's going to get a whole slide to itself. So the plasma membrane for neurons is just like the plasma membrane for any other cell in our body. It's that phospholipid bilayer. So you may or may not remember from anatomy and physiology or some other biology classes that a phospholipid has a phosphate head, which is hydrophilic or polar, meaning that it dissolves in water. So hydrophilic means water loving. It plays nicely with water. And then you have this lipid tail, and remember lipids are fats. Fats do not dissolve in water, so that lipid tail is hydrophobic or non-polar. It has no charge, therefore it does not play well with water. They stay separated. Now that phospholipid is arranged in a bilayer, so we've got our phosphate heads up here, phosphate tails there, and then sandwiched with another layer upside down there. So you've got the hydrophobic lipid tails in the middle of this sandwich, kind of like your peanut butter, and the phosphate heads are like the bread of a peanut butter sandwich. Now, That is going to become important because it's selectively permeable, as we're going to talk about in the next slide. Before we get there, though, I just want to point out that there are also proteins to be found in the plasma membrane. Some of them go all the way through from one side of the plasma membrane to the other, like you can see here. And so these are going to do lots of cool things for us. We're going to get to those later in the course. They can be channels and gates and pumps, and they do all kinds of really interesting things, getting things into and out of the cell. And they can do that because they cross from one side of the phospholipid bilayer, plasma membrane, one side to the other. Okay, let's talk about how it's selectively permeable. So this is so important for biology, and whether it's neuroscience or anatomy and physiology or whatever you're studying, the fact that the plasma membranes of our cells are semi-permeable is super duper important for then the cell physiology, or how cells work, and the things that can cause problems with them. So because you've got that peanut butter layer in the middle, which is hydrophobic, does not play well with water, the only things that can freely pass through in and out of the cell membrane, and therefore in and out of the cell, are small molecules. Like you can see here, we've got gases like oxygen and carbon dioxide. They can freely move in and out according to their concentration gradients, of course. And then also hydrophobic, right? So like lipid-soluble molecules like steroids. Now, steroids are a chemical class of molecules. Cholesterol is a steroid, estrogen is a steroid, testosterone is a steroid, cortisone is a steroid. So it's not just the anabolic steroids that you think of when we say steroids. Now, small polar molecules like water and ethanol, they have special channels that allow them to move through freely, so that's totally fine. But now, if we get to large molecules like glucose, it can't move in and out on its own, it needs help. And charged molecules such as ions, like sodium ions, chloride ions, potassium ions, calcium ions, things that are going to become really important to us when we're talking about cell signaling, cannot move through because they have a charge to them, so they're going to be repelled. They cannot play nicely with that central peanut butter hydrophilic lipid tail part of the plasma membrane. So, because ions cannot move freely, they have to be given special permission and they have to move in special ways. That means that we can control how much ion is on one side of the cell membrane. I'm using the terms cell membrane and plasma membrane interchangeably. They're the same thing, so my apologies if that caused confusion. Cell membrane is the plasma membrane. It's just a little faster to say cell membrane, and it also makes more intuitive sense. But anyway, okay, so the fact that those ions cannot move gives us an opportunity. So that's going to bring us to the unique properties of neurons, to number one of those, which is that neurons are electroactive. What does that mean? It means that at baseline, there's a charge gradient across their plasma membranes, across their cell membranes. The concentration of negative charges is higher inside the cell in the intracellular space. Intra means within. And there are relatively more positive charges outside of the cell in the extracellular space. So this is, you may have heard me, if you took anatomy and physiology with me, we refer to it as the resting potential. Technically, we call it the membrane potential because this can change over time, as we'll learn about in the class. But so the membrane potential is this difference in charge between the inside and the outside of the cell. And in most neurons, it's approximately 70 millivolts. Now, here's a little confusing thing. You see here, minus 70 millivolts. That's what MV stands for. Over here, for membrane potential, I have VM. I didn't come up with this, but V with a little m is the symbol for the membrane potential, or the difference in charge across the membrane of a neuron. Millivolts is the units for measuring the charge. Okay. Thing number two about neurons. They are forever cells. The neurons that you have today are the same neurons that you were born with, and you're not going to grow any new ones. So neurogenesis, or the ability to grow new neurons, there's debate around this. Maybe it happens a little bit sometimes under certain circumstances, but for the most part, it doesn't happen. So I found this really interesting study over here on the left, and they look at different types of cells and tissues. And what I loved about it was they included the germ layer. The germ layer is that ectoderm, mesoderm, endoderm that we talked about in neurodevelopment. So I thought it was really fun to remember those. And what they're telling us here is the estimated turnover. Now, most of the cells in our body are constantly being replaced. They die off and we make new ones constantly, all the time, but at different rates in different tissues. For example, the cells lining the inside of your intestine, where's my cursor? Here it is. So colon, which derive from the endoderm, right? Squishy bits inside, 3.5 days. Every 3.5 days, you basically have a brand new inside of your colon. That's pretty cool. So they're turning over really, really, really quickly. Most of the inside of your digestive tract is. You can see endometrium's right below there, 13 days, right? That's changing over quite a lot. Keratinocytes, which are skin cells, 64 days, right? And so you can see all these different cells have these different turnover times. And look, the longest one is neurons. Now, 32,000 days. Hard to really have a concept of what that is, so I divided it by 365 days in a year. That is 87 years, okay? So not in our lifetime are any of our neurons actually regenerating or making new copies of themselves. So neurons are forever cells. What you have is what you've got. You're not making any new ones, so please take good care of them. All right, so neurons are electroactive, they're forever cells, but they can change, right? Obviously, your brain today is not exactly the same as your brain when you were 1 year old. So there are structural changes that happen. There are also functional changes that happen. And so this is referred to plasticity. It comes from the Greek word that means being able to be shaped or molded. So it's the ability to change and adapt over time. There can be changes in the connections between neurons, the structure of the input sites that we're going to learn about. There is some limited ability to repair and regrow some neurons. It's not huge, but there is some. So we can have this kind of plasticity, and we know that lots of different things impact how our brains are organized, how our cells are structured and behaving, including things you've learned, things that have happened to you, things you've been exposed to. So what you've got is what you've got in terms of neurons, but there obviously is some room for change there, like for learning neuroscience. Okay, so those were some kind of functional properties of neurons that are unique. Now we're going to talk about some of the unique anatomical structures that neurons have. The first is dendrites. So dendrites are named that because it comes from the Greek word for tree, because they're like these little branches coming off of the cells. So these are the site of information input. This is receiving, I can't figure out, okay, receiving signals, it's mirrored, it's very tough. All right, so receiving signals through these branched things that are coming off of the cell body, which is often called the soma of the neuron. So some of those branches then have little protrusions, little spikes, little spines on them, which are sites of communication with other neurons. And so there's a lot of people currently researching how, like when memories form or when things happen to people, how those spines may actually be changing in response to that. So they're like, you know, a certain spine may contain a certain memory. I don't know, it's wild. Okay, so that's kind of cool. So we've got dendrites. Here's a diagram that shows the dendrites kind of highlighted in green. This is from the Hedges textbook. And then you can see they've zoomed in on one little area there to show you those little spines, those little extra projections on the dendrites to provide more opportunities for connections with other neurons. So then, of course, there's the cell body, which is also called the soma. We already talked about that. It has all the organelles. And then we come to the axon. So where the dendrites were the input, the axon is the output. And there's one single axon that leads out from the neuron. It can be very short. It can be micrometers, a little tiny microscopic, or it can be up to 3 feet long. So there's an axon that goes from the spinal cord in my lower back all the way down to my big toe. And it's just one axon all the way, right? So some are very short and some are very long. Now, there's a couple of interesting things about axons at each end. So you see right here, we've got the axon hillock. A hillock means like a little tiny hill. So this is a little kind of mounded part where we're moving from the cell body or the soma into the axon. And the axon hillock is responsible for kind of paying attention to all of the input that is coming from the dendrites and sometimes directly to the cell body or soma as well. Looking at all of the inputs, some of that is going to be stimulating the cells, some of it is going to be inhibiting the cells. So some of it is saying, send a signal, some of it is saying, don't send a signal. So we have these different types of input coming in, and the axon hillock is going to kind of weigh them. Do I send a signal? Do I not send a signal? And kind of decide. And if the axon hillock decides to send a signal, we're going to send a signal down the axon. And if it decides not to, then we're not going to. Now, if we do send a signal down the axon, it is going to travel in this direction to the end of the axon, which is the axon terminal. So a terminal, like a terminal illness, is the end of your life. A train terminal, it's like the end of the line. So a terminal, termination, you've been fired, right? A terminal means the end. So the axon terminals are these little, they look like those little frog feet that are at the end of axons, and they will release the neurotransmitters. So these little molecules or chemicals called neurotransmitters, which are going to be important for sending a signal. We're going to talk a lot about that next week. All right, a couple of other features about the axon. One is it's this big long projection, right? And sometimes you need to get stuff from the cell body down to the axon terminals, and sometimes you need to get stuff from the axon terminals back up to the cell body or soma. So you have these things called microtubules. You can think of them as like little railroad tracks, kind of like a little railroad system, and they can transport things back and forth along the axon. If we're moving from the cell body down to the axon terminal, that's called anterograde, because usually that's the directions things move, is from the soma down the cell body to the axon terminals. But if we're going backwards, that's called retrograde, back toward the soma. The other thing about the axon is we're going to have these little packets called vesicles. They're little packets of molecules. In the case of a neuron, they are neurotransmitters almost all of the time. And they're just these little membrane-bound, like little bubbles. Can I make that work? Okay, just little bubbles of molecules, little packages. We've put a little membrane around a group of them. And these can be released from the axon terminal. So here is it going to work? Yes. So here is a little animation of a vesicle that's going to kind of fuse with the cell membrane and release its molecules, in this case neurotransmitters, outside of the cell. So those are some additional features of the axon, the microtubules, which can do anterograde or retrograde transport, and then the vesicles. And then the piece de la resistance, the big important feature of an axon, is its myelin sheath. Now, not every single axon of every neuron has a myelin sheath. Some of the really, really short ones don't, but the longer ones absolutely do. So myelin is this fatty substance that's produced by special cells that we're going to learn about later in 2B. And it acts like an insulator, is the easiest way to think about it. So it acts like an insulator to allow for more rapid signaling down the axon. So this is so important because without myelin, especially on those like 3-foot long ones, it would take years for a signal to travel down the axon, which is like, that didn't help, right? So myelin is a necessary component in order to be able to send signals down an axon for the vast majority of our axons. There are some exceptions, but for most of them. Now, the myelin is not one continuous sheath down the entire axon. As you can see here, there are little spaces in between, and those are called the nodes of Ron-VA. And they're going to become important when we talk about how those signals move down the axon. So this myelin sheath, as I mentioned, is so darn important in order for most of our axons to be able to send signals at all. So if you have a problem or a disease that impacts your myelin, that can cause a lot of disruption. One example of a disease that does that is multiple sclerosis. Multiple sclerosis is an autoimmune disease. An autoimmune disease is a disease where your own immune system attacks yourself, autoimmune, right? Normally we don't. Normally our immune system is able to recognize, oh, that's one of my own neurons. I'm going to leave it alone. And then, oh, that's a bacterial cell, I'm going to attack it. So that's how our immune system normally works. But in an autoimmune disease, the immune system accidentally starts attacking some of your own body cells. And in multiple sclerosis, it starts attacking the cells that are producing and maintaining the myelin. So the myelin on the axon gets damaged, which results in the axons not being able to send signals. Now, what's very interesting about this particular disease is depending on which axons, which neurons, where in your body, you can get all kinds of different symptoms. So in the graphic here, you can see you might have problems with your vision. You might have problems with sensation. Sorry, my dishes are falling. You might have problems speaking. You might have problems thinking, right? You might have problems controlling your muscles or your bowel or your bladder. So depending on which particular axons of which neurons and which parts of your body are being impacted by this disease, you're going to have different symptoms, which can make it very difficult to diagnose, actually, because it's not always in the same places. So that's multiple sclerosis, a really important disease that is due to damage to the myelin. So we're going to finish out on neurons by talking about different types of neurons. So there's different ways to classify neurons. You can classify them by structure, which is their shape, or function, which is what they do. So for structural classification, it's going to be based on morphology. Morphology is the fancy name for shape of a cell, shape of anything, really. So like if you morph into something, that's changing shape, right? So morphology refers to shape. So there's kind of three basic morphology types of neurons. There's unipolar, means there's just one pole, so you have the cell body at one end and just an axon. Humans don't have those. Some invertebrates do, but we don't have those. We're too advanced for that. There are bipolar neurons where you just have one dendrite at one end, and then your soma, your cell body, and then you have your axon. And we don't have very many of those. We have some in the eye, as we'll see later. And then there are multipolar neurons, which are the ones that look familiar to you, right? That's the type of neuron that we are usually drawing or that you usually see in diagrams, and that's what most human neurons are. Now, there are lots of different morphologies. So we just talked about, bipolar versus multipolar. Well, these are some examples of other neuron shapes that we have in the human body. And so we'll learn some of these throughout the course, but just know there's lots of differently shaped neurons. But they all have a cell body or a soma, dendrites, and an axon, right? They're all going to have those structural features. The other way we can classify neurons is according to what they're doing. So what is their function? And we're going to usually break that down into three different types of functions. One is sensory, where the neuron is sending a signal out from the body up to the central nervous system, the brain and the spinal cord. Or if we're sending it in the opposite direction, we call that motor. So a motor neuron is sending a signal from the central nervous system down out to the body to tell it to do something usually. And then an interneuron is something that just exists in the central nervous system, and it's used to communicate between different parts of the central nervous system. All right, so that's neurons. We talked about their unique characteristics, most especially that they are electroactive with that membrane potential of -70 millivolts. We talked about how they're forever cells. What you've got is what you've got, but they do have plasticity. Then the special anatomic features of these cells with the dendrites that have spines, which are the site of input. Then we've got the cell body or the soma, which has all the usual organelles. And then the axon, which has an axon hillock, which is kind of the decider. And then we go out to the end, the axon terminal. We've got microtubules, that railroad system for moving things either anterograde down toward the axon terminals or retrograde back toward the cell body. Vesicles, little packets of neurotransmitters. And then the axon itself is wrapped in a myelin sheath, but there are little breaks in that called the nodes of Ranvier. And then we talked about the clinical example of multiple sclerosis, which is a disease that attacks myelin. We then briefly talked about different ways of classifying types of neurons. So the structural or morphology-based classification, there's bipolar or multipolar, and then there's other names that we'll learn based on some more specific shapes, and then functional classification. So is it sensory? Is it sending information from the body up to the CNS? Motor, sending information from the CNS out to the body? Or is it an interneuron, which is just within the central nervous system? So that's neurons. We'll pick up with part 2 of lecture 2, talking about neuroglia. See you then.